So in my lab, we're very much interested in how the brain and body communicate in mental health disorders. And what we're always trying to identify are conduits between the body and the brain that might be influencing behavior. And so really, whenever we're studying the the role of the role of the immune system in modulating the brain, we necessarily have to look at both systems. And so my lab originally began this work by thinking about what psychedelics are doing to behavior. And we also happen to be profiling the immune system at the same time, just to see, more or less as a control, that nothing really would be changing, because we essentially thought psychedelics were acting neuron autonomously. But we were very surprised in some of our original pilot experiments that it actually had these modulatory effects on the tissue, where actually um, the number of immune cells in the brain tissue was reduced by administration of these psychedelics. And later we worked out, a more detailed mechanism by which this might be happening. Um, and so the, the inspiration for doing this in the first place is really coming from the fact that in mental health disorders, we've been so focused on what's going on in the brain for such a long time, and rightfully so, um, that I think we neglected a little bit of what else could be modulating brain function, namely these peripheral cells that are dynamically circulating throughout multiple tissues, including the brain. And so we're really interested in my lab in using immunotherapy as a potential treatment for mental health disorders. So the relationship to us is that in response to chronic stressors, there's kind of this liberation of immune cells from different reservoirs in the body, and they eventually find their way towards tissues adjoining the brain, or even in some cases moving through the brain itself. Um, and when they're there, they're not just bystanders. They actually are actively releasing certain molecules. And so what we found is this link between cytokines produced by these cells, which are essentially signals that are released by immune cells, that can actually penetrate deep into the brain in the stressed mice. But in normal mice, they don't penetrate very deeply at all. And the reason for that is because there's kind of this more open barrier that's normally closed in the depressed, or rather the stressed mice, that sort of lets these in. And when these cytokines get in, they can really start to act on cells in the brain that aren't used to seeing very much of these molecules. And when that happens, it actually physically appears to be stabilizing contacts between different types of brain cells that normally aren't in such strong contact. And this actually leads to increased brain activity in certain areas. And so in our case, we focus on the fear center. And so when you have greater stabilization of fear responses, it kind of leads to these behavioral symptoms that we observed. And so the relationship between the monocytes and EGFR and the fear is that the monocytes themselves are trafficking to a tissue layer that surrounds the entire brain, really the entire CNS, called the meninges. And it's there that they can release some of these factors that can get through the impaired brain barriers. And what happens is it actually appears to be down regulating the expression of EGFR, which sort of acts like a brake, an anti inflammatory brake within the brain in certain types of cells. And when that EGFR signaling is down regulated, it leads to this upregulation of molecules that stabilize contacts with neurons that promote fear. And so that's kind of the model for how we think this might be working. NR2F2, we found, is a transcription factor that is expressed in neurons of the amygdala. And what it appears to be doing is activating machinery that induces the formation of excitatory synapses. And when this occurs, it actually leads to increased excitatory drive from the amygdala, meaning that there's increased fear. Um, and so NR2F2 fits in because we discovered this interplay between glial cells in the amygdala and neurons in the amygdala. And when EGFR signaling is dysregulated in the glia, it ultimately leads to increased transcriptional activation in the neurons of NR2F2, which essentially leads to a fear stabilizing program transcriptionally. And we think it's partly due to inducing machinery that facilitates synapse formation. So basically just making these neuron-neuron contacts to convey more fear to other parts of the brain. We were really interested in how psychedelics modify stress behaviors related to mental health. And so that's why we were testing them. We just happened to originally be studying basically the distribution of immune cells in stressed mice, and we tried them in these experiments just to see if there were changes in behavior first of all, and then also we were examining, what are the immune populations that are changed by stress, and we just happened to have the psychedelic treatment, and so we were we were very surprised because what we found is that there is this redistribution of immune cells after treatment with psychedelics. And so that's kind of how we discovered it. And, looking into the literature more, we found that there's actually a basis for their activation of non-neural cells because they act on receptors that are widely expressed throughout the body. And there's been previous reports that psychedelics may be modulating inflammation in a beneficial way. And the specific psychedelics we were using were psilocybin, which is a compound from magic mushrooms, and also MDMA, which is called ecstasy. And we were using them because they had clinical efficacy in trials for depression and PTSD. And so we thought they would be among the most clinically relevant. And also because in recent studies in animals, they were shown to potently control behavioral phenotypes linked with social reward and also stress behaviors. And so that's how we settled on those two. But now we're very interested in a whole host of psychedelic compounds and whether or not they can do the same sort of thing. Yeah, so the way we view this whole mechanism working is what we found is that in the mice that were chronically stressed and showed behavioral deficits and immune cell accumulation, they actually exhibited vascular dysfunction, like I mentioned earlier, kind of these leaky barriers in the brain. And we could visualize this with a fluorescent dye. But what happened is that when we administered psychedelics, that vasodilation, for lack of a better word, was reduced both by MDMA and psilocybin, suggesting that the psychedelics are actually inducing vasoconstriction. And so that, coupled with the fact that we detect simply more immune cells that are housed in the tissue layers adjoining the brain, suggests to us that in the stressed mice and potentially in depressed patients or patients that exhibit similar symptoms, there might be this increased burden of immune cells because due to vasodilation, and then the psychedelics can reverse that by sort of constricting the vasculature and squeezing the cells out so that they essentially redistribute elsewhere. And we partly think this because actually we were able to block the effects of psychedelics in the paper by administering to mice a systemic vasodilator, and so it boosted the vasodilation even further. And then we saw actually more cells even in the presence of psychedelics. So what we think is happening is this multicellular circuit that's recalibrating tissue homeostasis. So for us, if the mechanism is related to neurons interacting with vascular cells, interacting with immune cells, we think that there's actually a very big potential for these compounds in multiple diseases because thinking about across the entire body, almost every single organ is innervated by the nervous system. It is it it immune cells circulate to these organs, and there's structural cells and vascular cells that are in these same tissues. And so we think that this might be a general principle of tissue homeostasis with certain windows of opportunity to use these drugs. And so we're very excited about that. And so we're so in the lab side, we're really interested in determining whether or not these drugs can influence inflammatory diseases that we can induce in the animals. Um, Clinically, we're collaborating with researchers at MGH running a clinical trial of psilocybin and depression. We're trying to understand whether or not these changes in immune cells are happening in patients, and whether they're as long lived as the behavioral modulation. Um, and I think that that's kind of really where we're excited to go, is to see how broadly this principle can be applied.